Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, so let's dive right in. Imagine this for a second.
Yeah.
One of your cells, it has a bit of a rough day.
Right.
Maybe just, a single hiccup while it's copying its DNA, what researchers call replication stress.
Yeah.
You'd probably assume, okay, fine, the cell fixes it, divides, and the two new cells are, back to normal. Good to go.
And that's where this deep dive delivers, well, a real surprise. Because this new research we're looking at shows that's often not what happens at all.
Oh.
That single event, even if it's transient, it can leave a lasting, a scar. Not just on that cell, but it impacts its descendants too.
And here's the bit that really got me the counterintuitive part.
Yeah.
That stress from the parent can actually make the two sister cells, the ones born from that division, make them behave completely differently from each other. Sometimes even generations down the line, they start, diverging, even though they came from the same parent cell.
Exactly. It's fascinating. We are digging into a really groundbreaking paper from Nature. It's titled Multi-Generational Cell Tracking of DNA Replication and Heritable DNA Damage.
Okay, so our mission today is to unpack the core findings here. We need to look at the seriously clever technology they use to see this stuff.
Which was previously pretty much invisible, yeah.
Right. And understand what this means for how cells handle damage over time and, why this really matters for thinking about cell health and disease.
Absolutely. So just to set the stage a little bit, it's really important to remember that the cells in your body, they aren't all perfect identical clones.
Right. There's natural variation.
Exactly. Cellular heterogeneity, that's the term. And this isn't just background noise. This variation can be incredibly important, especially when you think about diseases like cancer.
How so?
Well, in a tumor, for example, different cells can have wildly different properties, affecting how they grow, whether they spread, how they respond, or don't respond to treatments. Heterogeneity is key there.
Okay, and where does this variation, this heterogeneity come from?
So one major source can be problems during DNA replication, or, just straight up DNA damage from various sources. Cells have these really complex systems to try and fix these issues. And a key protein involved in spotting certain types of damage, particularly DNA double-strand breaks, is called 53BP1.
53BP1.
Yeah, think of it like a specific kind of first responder. It gets called to the scene, to the broken DNA strand, and helps coordinate the repair.
So cells get stressed, damage happens, 53BP1 jumps in. Got it. But the big question this paper tackles seems to be about the long game, right? The fallout later on.
Yes, exactly. We've known for a while that cells can inherit DNA problems. These lesions can get passed down. But what's been really murky is how these inherited problems actually affect future cell cycles.
And specifically,
whether they cause differences between the two sister cells that come from the same division. Do they both inherit the same baggage equally? Or is it somehow split unevenly, leading them down different paths?
that's the core question. And figuring that out sounds incredibly difficult. How on earth do you watch individual cells track their kids and grandkids, and see the specific damage inside them? Cells move around. They all look kind of the same.
And that is precisely where the innovation in the study shines. If you look at their figure 1, they developed some really, really cool technology.
Okay, tell me about it.
They used CRISPR-Cas9 genome editing to engineer human cells. They tagged key proteins with fluorescent markers, but crucially, they did it endogenously.
Endogenously, meaning?
Meaning they modified the cell's own genes, so the cell itself produces the tagged protein right where it's supposed to be as part of its normal machinery. It's not just sticking a tag on from the outside.
Okay, so the tag is built right in. Much more natural.
Much more natural. And they tagged 2 key things. 53BP1, our DNA damage responder, they tagged that with a red fluorescent protein.
So damage sites glow red.
Exactly, I'm Scarlet. And they tagged PCNA, that's a protein that's essential for DNA replication. It kind of clamps the copying machinery onto the DNA. They tagged that with a green one, emerald.
Okay, red for damage, green for active DNA copying. How does that help track generations?
Well, the beauty is, especially with the PCNA signal, which changes its pattern throughout the cell cycle.
Uh-huh.
It allows specialized software to identify each individual cell nucleus in the microscope images, frame by frame, over long periods. And by watching which cell divides into which two daughters and tracking when and where those red 53BP1 spots appear, the software can build these incredibly detailed lineage trees, basically cell family trees showing exactly who came from who inherited what damage, across maybe 4 or more generations. You can see it at figures 1A and 1B.
Wow, so you get a whole family history mapped out with damage events.
Precisely. And they did their homework, checked carefully that adding these tags didn't mess up normal cell behavior or division speed. That's figures 1C to 1E. And, really importantly, the changing pattern of that green PCNA signal allowed them to figure out the exact cell cycle phase, G1S phase or G2, for every single cell in every frame of their movies. That's figures 1F through K.
And why is knowing the phase so critical?
Because damage happening in G1 before DNA replication might have very different consequences than damage during S phase, or damage that happens in G2 after replication is done but before division. Timing matters.
Okay, that makes sense. And they added another layer too, this live plus QIBC thing.
Yeah, that's pretty clever. It's shown in extended data figure 4. Basically, they do the long-term live imaging, tracking the cells and their lineages.
Right.
And then at the end, they fix the very same cells on the dish and stain them for other important proteins using antibodies. Things like H2AX, another DNA break marker, P53, the tumor suppressor, and key cell cycle regulators like P21 and PRB.
So you connect the movie of their life story with a detailed molecular snapshot at the end.
Exactly. You link the dynamic history, when damage appeared, how division happened, with the state of all these other important molecules in those exact cells. It's like watching a family grow up on film and then giving each member a detailed medical checkup, linking the two.
That's incredibly powerful. Okay, so they've got this amazing tracking system. What happens when they actually introduce some stress? This brings us to figure 2, right? The emergence of sister cell differences.
Yes, this is really the heart of the paper, and the visuals are striking. By tracking thousands of sister cell pairs born from the same parent cell that experienced stress, they could directly compare how those two sisters behaved side by side and see them start to diverge.
What kind of stress did they use?
They used mild temporary replication stress, either a drug called aphytocolin, APH, which slows down the DNA copying machinery, or an ATR inhibitor, ATRI, which blocks a key protein involved in responding to replication problems. They treated the parent cells for just a limited time, washed the drug out, and then just watched the cells and their descendants for several days.
And compared to normal unstressed cells, the difference was?
Dramatic. Look at figure 2A and 2B that shows normal untreated cells. As you'd expect, sister cells are highly synchronized. They enter S phase at roughly the same time. They have very low damage markers. They generally act like identical twins.
Predictable. Makes sense.
Right. But then look at figure 2C. This is after the parent cell got just a brief pulse of APH stress while it was in G2. The immediate daughter cells, they might look okay, maybe divide normally, but then the granddaughters,
2 generations later,
they start showing higher levels of DNA damage markers like H2AX and P53, but not always both sisters equally. And crucially, you see sisters starting to differ in their cell cycle control. Some show high levels of P21 and low PRB.
Those are the brakes on cell division, right?
Exactly, the brakes. So some granddaughters have trouble committing to enter the next S phase, while their own sister might be fine and proceed normally.
Wow. So a temporary stress 2 generations ago leaves this legacy where today's sisters are now out of sync regarding damage and division.
Precisely. And it gets even more pronounced with the ATR inhibitor stress applied in the parental G2, that's figure tussin. In that case, the sister cells, right after they're born, immediately show big differences.
Like what?
Differences in how long their S phase takes, differences in whether they have those 53BP1 damage foci, differences in activating stress markers like H2AX and P21. They are immediately behaving differently, dealing with the inherited situation in distinct ways.
This is huge. So a brief stress in a parent cell doesn't just cause a temporary problem. It leads to persistent damage, and critically, it actively increases the variability between sister cells. They start as siblings, but quickly take different paths because of something their parent experienced.
It fundamentally changes how we might think about cells remembering stress. It's not just a uniform memory, it's passed down in a way that actually generates asymmetry between the daughters.
Okay, so they've clearly shown that stress causes this sister cell heterogeneity. Which leads straight to the next big question. Why? Why do sister cells coming from the same parent with essentially the same DNA start behaving differently? What makes them diverge? That's figure 3, right?
Exactly. They needed to dig into the sources of this divergence. Is it just random fluctuations in gene expression, or are there other, maybe non-genetic factors at play?
And what did they find?
Well, they looked closely at that protein we mentioned earlier, 53BP1, the damaged first responder.
Yeah.
And while the overall amount of 53BP1 in the whole cell population didn't change drastically after damage, they found something interesting at the single cell level. The actual amount of 53BP1 protein just floating around inside the nucleus varies quite a bit from one cell to another. You can see that in figure 3.
So even in a group of seemingly identical cells, some just happen to have more of this key protein readily available than others.
That's right. And this individual difference really matters. Figure 3 shows that cells starting with lower levels of nuclear 53BP1 are actually less effective at forming those visible damage foci when they encounter new DNA breaks, like from radiation.
Ah, so if you don't have enough responders ready, you can't even signal the alarm properly.
Kind of, yeah. And figure 3e shows examples in actual cell lineages where sister cells inherit slightly different amounts, and the one with higher 53BP1 forms more damage foci in response to radiation than its sister with less.
So your starting protein level dictates your visible response. That's one source of difference.
That's one. And figure 3f adds another really interesting layer. If a cell already has some 53BP1 tied up, dealing with damage from previous replication stress, often forming these dense clusters or condensates in the G1 phase, that 53BP1 is essentially sequestered. It's busy.
Right.
Which means there's less free 53BP1 available to quickly respond if new acute damage happens somewhere else in the genome, like from radiation.
So old problems can hinder your ability to deal with new problems, like the emergency services being tied up at one incident, making the response to a second incident slower.
That's a great analogy. Now, think about sister cells again. After division, if one sister happens to inherit a bit more free, available 53BP1, or maybe the old damaged sites with sequestered 53BP1 aren't divided perfectly evenly between them, then those two sisters are immediately set up to respond differently to any new stress they encounter. So this non-genetic factor, just the amount and availability of a key protein, can significantly contribute to them taking different paths.
And they also showed that even a single hit of damage, like one dose of radiation in G1, could ripple forward and cause noticeable differences in the next generation, affecting when they replicate and divide. That's in the extended data, right?
Yeah, extended data figure 8. It really hammers home the point. Even a brief, acute hit can leave a lasting, heritable difference that plays out as heterogeneity between descendants.
Okay, so stress causes sister cell heterogeneity, influenced by things like 53BP1 levels and existing damage. What are the big downstream consequences of this divergence, beyond just, cell cycle timing?
Well, one really critical outcome they investigated is polyploidization.
Polyploidization, meaning?
Meaning cells end up with extra sets of chromosomes, more than the normal 2 copies of each. This is something often seen in cancers, particularly aggressive ones, and it's frequently linked to drug resistance.
Okay, so cells getting extra DNA, how did they study that?
Using their lineage tracking, they could actually watch individual cells become polyploid in real time and figure out how it happened. They looked at cells treated with a drug called pevonedistat, which is known to sometimes cause this. And they also looked at cells they'd engineered to overexpress common cancer-promoting genes, oncogenes, like HRAS or cyclin E1.
And did they find just one way cells become polyploid?
No, and this is really interesting. They identified 2 main pathways, 2 distinct routes that these stressed cells took to become polyploid. It's shown nicely in figures 4e and 4f.
Okay, what are the routes?
One route is re-replication. This is where a cell messes up and starts copying stretches of DNA that it already copied earlier in the same cell cycle. It looks like chaotic, uncontrolled DNA synthesis, as you see in figure 4c. It's like trying to rewrite a chapter you just finished, but doing it sloppily within the same writing session.
Got it. Flawed copying within a cycle. What's the other way?
The other way is endoreplication, sometimes called endocycling. In this case, the cell actually completes one, or sometimes even multiple full rounds of DNA replication fairly accurately.
Okay.
But then it completely skips cell division. It doesn't go through mitosis or cytokinesis. It just goes straight into another S phase, but now with double or quadruple the DNA content. That's figure 4a. More like finishing a chapter perfectly, but then immediately starting the next chapter without properly closing the book or turning the page.
Okay, so re-replication is messy copying during S phase, while endoreplication is skipping division between S phases.
Exactly. And here's a crucial finding. The route the cell takes to get that extra DNA matters a lot. Figures 4G through K show that the cells that became polyploid via re-replication,
The messy route.
the messy route, yeah, they accumulated significantly more DNA damage markers, more gauge2AX, more p53 activation, more p21 compared to cells that became polyploid via the endoreplication route, even if they ended up with the same final amount of DNA.
Wow. So re-replication seems inherently more damaging, more stressful for the cell than endoreplication.
It certainly looks that way. The process itself seems to generate more problems, and they also showed in Figure 5A and the related extended data that those common cancer genes, HRAS and cyclin E1,
Yeah.
they also ramp up the cellular heterogeneity and push cells towards polyploidy using both of these routes, re-replication and endoreplication. It seems like oncogenes can exploit these pathways to generate abnormal cells.
And did the timing of the original stress influence which path the cell took?
It did. They found, this is an extended data figure 12C, that damage experienced early in the cell cycle, like in G1, tended to increase the risk of re-replication happening in the next S phase.
Okay.
Whereas damage occurring late in the cycle, in G2, increased the risk of the cell skipping mitosis and undergoing endoreplication instead. So the cell state, when it gets hit, influences its future abnormal trajectory.
This whole study just seems to be pushing the boundaries of what we can even see inside living cells. What do you think is the biggest innovation here, and how does it maybe inspire future work?
I think the biggest innovation, hands down, is combining that really long-term, multi-generational live cell tracking with the ability to do detailed molecular analysis on the very same cells afterwards.
Right.
Before this, we were mostly stuck looking at averages in cell populations or maybe tracking cells for just one division if we were lucky. This approach lets you follow thousands of individual cell life stories and see exactly when, where, and how differences emerge within a family tree.
It really is like shifting from looking at, I don't know, population census data to watching detailed biographies of individual families unfold over generations.
That's a perfect analogy. It allows us to really get at the origins of variability, not just observe that variability exists. And the implications are pretty significant.
How so?
Well, it fundamentally changes how we think about cellular heterogeneity.
Yeah.
It shows it's not just some inherent static noise in the system. It can be actively generated by things like transient stress leading to stable, heritable differences between cells.
And why is that so critical for understanding disease?
Because in diseases like cancer, cells are under constant stress from the environment, from treatments, from their own rapid growth.
Yeah.
This ability to induce heterogeneity could be a powerful engine driving cancer evolution.
Ah, I see.
You could have sister cells, born from the same parent tumor cell, immediately start going down different paths. Maybe one accumulates more damage via re-replication, another becomes polyploid via endoreplication, maybe one becomes resistant to a drug while the other doesn't. This constant generation of diversity within the tumor makes it incredibly hard to treat effectively.
It provides a mechanism for how resistance could pop up so quickly, right?
Yeah.
Even if the tumor seemed uniform initially.
Exactly. And it changes the kinds of questions we need to ask. It's not just how does the average tumor cell respond anymore.
More like.
It's more like how do individual cell lineages evolve within the tumor? What makes one sister cell more likely to survive treatment than the other? And maybe even, can we target the process that generates this dangerous heterogeneity? Can we specifically block re-replication, for instance?
That opens up completely new ways to think about therapies, doesn't it? Targeting the evolution itself.
It really does. It forces us to see the cell not just as a generic blueprint, but as an individual with a history, carrying baggage from past events influenced by subtle things like how much protein it happened to inherit.
So, wrapping up this deep dive then, what's the absolute core message you'd want people to take away?
I think the core message is that transient DNA damage or replication stress isn't just a momentary blip that gets fixed and forgotten. It can leave a persistent, heritable mark, a scar that actively increases heterogeneity within a cell population, specifically causing sister cells to diverge in their behavior and their ultimate fate, sometimes across many generations.
And this divergence isn't trivial. It can manifest in different ways affecting the cell cycle, how accurately DNA is copied, and even pushing cells down distinct pathways towards dangerous states like polyploidy, with each pathway carrying its own risks.
Right. Thinking about cells as individuals whose future is shaped not just by their genes, but by the inherited consequences of past stresses and these non-genetic factors, it adds a whole new layer of complexity, doesn't it?
It really does. It changes the whole picture. And it makes you wonder, how might this deeper understanding of individual cell fates and where this variability comes from change how we tackle diseases like cancer that are fundamentally driven by this kind of cellular evolution?
Yeah.
And, what other seemingly minor stresses might be leaving these lasting unseen marks on our cells throughout our lives?
That's a fascinating thought to end on. Definitely gives us a lot to think about.